I'm David Craig. I'm a cancer researcher, and I work with a team of researchers at the University of Vermont and other institutions in the United States and outside the United States. Our goal is to improve treatments of cancer by using a person's own immune system to fight their own cancer. Now, the rationale for this is that we know that people develop an immune response to their own cancer. The problem is that that response just doesn't go far enough, but it's there, it starts. And our goal is to boost that way up and make the cancers go away. Important components of the immune system are the lymphatic vessels themselves and lymph nodes. Now, the lymphatic vessels are located throughout our body. It's a vast network of tiny little vessels that are constantly absorbing fluid that's the clear tissue fluid that percolates throughout our whole body. They're kind of like a hose sucking groundwater out. And they collect that fluid and they bring it and collect it all the way back, ultimately to be returned into the blood system. That occurs right about here, about 4 or 5 inches from the heart. And there's a lymphatic vessel that communicates with a major blood vessel, and that's where it dumps back in. But it comes from that vast pool that circulates throughout our whole body. Now, stuff gets into our tissue fluid. We'll get a bite from a cat, or we'll get a thorn or a small infection or cancer. And those things get into that groundwater and get brought into the lymphatic vessels and get taken to lymph nodes. Now, the reason that they pass through lymph nodes is that we wouldn't want to have bacteria and all kinds of absorbed crud just float from the lymphatics back into our blood. So nature devised lymph nodes, and they act like a filter as the incoming fluid comes in to trap it, and importantly, to start to develop an immune response against that foreign material. Lymph nodes are located in clusters, like in our armpit or our neck area or in our abdominal cavity. And in the armpit area, there's an average of about 15 of these things. Now, what goes on inside that lymph node is the very first initiation of the immune response to either a tumor, breast cancer, melanoma, prostate cancer, or a vaccine. In comes that material, and the lymph node starts to process that with special cells, which include both B cells, T cells, and other cells. We're going to focus in on the B cells, because the B cells are the special cells that make antibodies. Antibodies are special molecules that are released from B cells. Like when you blow on a dry dandelion, off those little floating seeds go, and the dandelion is left behind. And so a B cell produces those to secrete and release antibodies that get into our bloodstream. And a B cell that's really pumping out can pump out up to 10,000 of these per second. The clear part of our blood has a lot of antibodies in it, all kinds of antibodies that our body's B cells have made over the years, and they stay circulating for weeks and weeks and weeks, waiting to defend ourselves against whatever. So, for example, you get a cut, you get an infection, maybe you're in an emergency room and they think maybe you need a tetanus shot. Well, you can't remember when you had your last tetanus shot, so they'll test your blood to see if you have the antibodies against tetanus toxoid. And they check your titer. What they're really looking for is to see if the antibody level from that past vaccine is still at such a level that there's enough antibodies in your blood to attack the tetanus toxoid. If not, you get a vaccine, and the process is repeated and boosted, and more B cells then are stimulated to make antibodies. Well, we want to do the same thing for cancer, basically. But it just doesn't do it. Not enough, anyway. So I developed an several years ago a technique that allows surgeons to precisely identify the lymph node that's receiving drainage from a lymphatic vessel. So for example, in breast cancer, we want to know what's the first lymph node that's receiving drainage from that cancer. The way that's done is to inject a radioactive tracer into the breast that gets absorbed into those lymphatic channels in the clear tissue fluid and carry that wherever it goes to the first encountered lymph node. That lymph node traps that tracer, making it slightly radioactive, and the surgeon with a handheld detector can go back and forth sensing where that is, and it makes an audio feedback till the surgeon can point exactly at that lymph node before ever making an incision. It's just right there. The same can be done with an injection of that radio tracer around a vaccine. If we got a flu vaccine, I could inject that tracer next to the vaccine and I could find the precise lymph node receiving drainage from that vaccine. So our goal going forward is to take a vaccine, a cancer vaccine, and to remove the lymph node that's draining that exact site. Now, why would we do that? Well, we know from our experience and our research two very important things. Lymph nodes that receive drainage from a cancer do have those special antibodies inside them, inside the lymph node, that secrete the antibodies that attack cancer. We also know in patients that have been vaccinated with a melanoma vaccine that those B cells are in that lymph node receiving drainage from that vaccine. Now, this is a very beautiful thing. This is a very important aspect to the research, because once we get our hands on those B cells, our next goals are to then, in the lab, grow up cells that produce those antibodies to a high enough level that we can then give those back to the patient. Now, should that be a safe or a dangerous procedure? We expect it to be a relatively safe procedure because we're giving back to the patient their own antibodies. They're already floating around in the blood in that patient. They're just not at a very high level. So we boost the dose way, way up. So we're heading in that direction that we should be able to achieve this in the near future. Kat Bordy, Linda Craig, and several of their colleagues have established a network to try and help raise the funds to help us speed this whole process along. Our lab research team is very clued into their whole efforts to do this and are very appreciative because we know how important it is to help provide the resources to make this research go forward. So we're very appreciative of this to help us along our goal to try and speed this to clinical studies. And we want to thank you very much. So I'm here on an exceptionally warm day in Vermont. It should be snow on the ground, which it was a few days ago, but here it is, warm as can be. People are the goals of what we're trying to help, and it's all personal. And I think it's not some sterile environment in a hospital we're stuck in. We all live in different places and we all need to connect together, and the more we do that, the quicker we'll be able to solve this problem. So thank you again.